Right, so today we'll be starting off with a continuation of where we left last time. We left on the components of the cell membrane. So we had done only glycolipids and phospholipids in a bit detail, but now we'll be starting off with the proteins of the cell membrane, another important component of the cell membrane. So first I'll explain the types or the two major types of proteins present in. So there's intrinsic proteins and there's extrinsic proteins. As the name suggests one is in the internal part of the cell membrane and one is on the external part, right? So extrinsic proteins are only half span, so like they're present on the outer monolayer, right, like this. So this can be an extrinsic protein, right? However, for intrinsic protein, there's full span and there's a half span protein. So the major focus on our syllabus is a full span protein. Half span proteins and extrinsic proteins are just mentioned. So I'll just explain how they look. This is a full span protein, and right. So this is a full span protein because it covers the entire bilayer, and it is usually used for transport that I'll be telling on later on. An intrinsic half span protein is either on the outer or inner monolayer but present internally, right? So this is an intrinsic protein but half span, right? So as I mentioned, full span is what we'll be majorly dealing in. I'll show you a picture right here. These right here that you see, these are integral proteins or intrinsic proteins as they're present internally. However, this one right here is an extrinsic protein, and these two, right, these two are what our syllabus majorly focuses on. This is what we'll be majorly doing, and these are called transmembrane proteins alongside full span proteins as well, because they're involved in transport and they're present like throughout both the membranes, right? So they're transmembrane, and because they're transmembrane, they can be involved in transport, and I'll tell you how they're involved in transport. Right, so we've done this part, and how now our major focus will be on transmembrane proteins only. So we can give the heading of transport proteins. Now, even in transport proteins, there are two types of proteins. The first type is channel proteins, and the second type is carrier proteins. We'll leave aside carrier proteins for a while. We'll focus on the channel proteins for now. So what a channel protein is as you've seen, this is a monolayer and this is another monolayer, right? So these monolayers, and this is a full span protein. So this protein right here, it is a channel protein. Now I'll tell you what a channel protein is in a while, but first let me tell you that the outer covering, the one that I've marked with yellow, is always going to be non-polar, because the major part of the cell membrane is non-polar, as we've studied earlier. So to be accommodated internally, for accommodation, these proteins are always non-polar. However, there is this very narrow channel that I've marked with white, this channel right here. This channel is a polar or a hydrophilic channel. Now we've learned hydrophilic means polar, and it is a water filled channel, right? So it helps those polar substances that we couldn't transport earlier to get transported internally. Like for example, glucose can travel through this, mineral ions such as sodium ions, potassium ions can travel. So the sodium ion go through this polar, very thin polar region, and if you can see in this diagram as well, as I've mentioned right here, right, so this is a channel. Through this channel the polar substances can pass through. However, this is still a non-polar protein. So the two things that we have to know is that this channel protein is a fixed shape, that is, it doesn't move It doesn't show a conformational change that other proteins do. We'll study them later on. And it is specific to one type of ion. So for example, if this protein uh, transports sodium ions, so it will be a sodium carrier protein, right? So Na plus carrier. If something transports glucose, that will be a glucose carrier, and further on, right? So this is what a channel protein is. It has a thin channel going through the bilayer, and it is non-polar on the major part, but because it has to be accommodated inside. However, this thin polar region is through which all the polar travel through. As we've studied that polar can attract polar and non-polar is rejected by or repelled by polar always. Now, another type of channel protein is a gated channel. Now, what a gated channel is, it has a gate to control the movement. It has an almost similar shape, but it just has a gate to control. So, for example, I'll make one right here. This is the gate that's controlled. This gate opens up, and a specific ion travels inside. Right, so not a major difference between the gated channel and the normal channel proteins. However, the second type, carrier proteins, that we'll study next is a major difference. Right, so we have to discuss carrier proteins now. So first of all, I'll tell you the types of carrier proteins. Right, so just like the previous ones, the channel proteins, even carrier proteins have two types. One is the simple carrier protein, and the other one is pumps. Right, so we'll discuss what each one does. So first we're discussing the main carrier proteins. So there's not much of a difference for the carrier proteins as opposed to the channel proteins, but it does have a similar shape. However, instead of the channel protein having a small channel through, this one carrier proteins has an entire shape change to get its substances inside the cell membrane. Right, so I'll explain how it does that. It undergoes a conformational change. What a conformational change is, an inversion in its 3D shape. Now these are just words. I'll practically demonstrate how they're done. So how they undergo conformational change and an inversion in 3D shape is shown below. So I'll show it right here. Right, so this is a carrier protein, and if you see right here, I'll make right, so think of this as a glucose molecule. Right, so this glucose molecule comes and binds to the specific binding site for glucose. Now it binds right here, and because of its specificness, as we've learned, specific, so one type for one molecule or one ion, it attaches right here and it inerts fully, as we've established earlier, that it shows an inversion in the 3D shape. So it shows an entire inversion to become this, right? And just like this glucose molecule is now inside the cell membrane, and after it takes the glucose molecule inside, it has to get back up to take more glucose molecules. So it everts itself to the initial shape and takes more glucose molecules, and then it goes inerts, and the cycle continues. Right, now this thing that it does is called a conformational change. This it does not invert instantly, as I've shown right here. It goes through a process like peristalsis that we've studied uh, like a bolus travels through the esophagus using smooth muscles and all. So this is how it actually travels. This is the pathway it takes, right? And as it travels down, it changes its shape from the specific binding site here to the specific binding site here. And because it is diffusion, it is not active transport, it does not need energy, it's done passively, right? So this is a carrier protein. Now the second type of carrier protein is a pump, as I've mentioned earlier. Now the main difference for a pump is that it is used for active transport, right? So and as it is used for active transport, it requires energy, and energy obviously comes from ATP, because ATP is the energy source in all living cells. It is the currency, as we can say, for energy. ATP, adenosine triphosphate, right? We'll learn about that later on in the future chapters, but for now you need to know that energy comes from ATP. So right, so because it's active, it moves against the concentration gradient. So this is an example of how carrier protein with a pump is shown. They haven't shown the exact active sites and the specific binding sites for the molecules that should be present, but they've just given you an idea of how ATP hydrolyzes to ADP and pi. I'll explain that as well. So look, so this is a similar diagram I've shown earlier, in the carrier proteins, but this has another binding site right here. This one is for ATP, and this one is for any molecule that attaches right here. It could be a potassium ion, a sodium ion, a glucose molecule, anything of that sort. But what we have to remember is that it's against the concentration gradient. So this means they're fewer in number here and they're more in number here. However, they still travel from a concentration, from a lower concentration to a higher concentration, right? So this ATP binding site, an ATP molecule comes, it hydrolyzes into ADP and pi. What pi is a phosphate molecule. We'll learn that later on. So as I've stated here, ATP hydrolyzes into ADP and pi, providing energy. So if anything is hydrolyzed, so energy is given out, right? That energy is used for the conformational change to bring in molecules, right? So it changes the entire shape and it brings in the specific ion, for example in this case sodium ion, and that is because of the energy that's released through ADP and pi hydrolysis. Now why energy is required is because it's an active transport process. It requires energy. It's not passive, unlike diffusion. So this is what a pump is required for. Now these all the different active transport in the cell membrane, for example, always require pumps. Always active transport takes place through pumps, and for example, for like facing cytoplasm is always an ATP site, and facing the outside of the cell membrane is always a site, for example glucose, for sodium ion, for anything, and we call it a glucose pump or a sodium ion pump, for example Na plus pump, right? And this is where we'll conclude our video for today, and we'll get on with glycoproteins in the next video. I'll just do a quick recap. What we started off was with the proteins of the cell membrane, the types of proteins. So there was intrinsic, extrinsic. There were two types of intrinsic. The main focus on intrinsic is full span, because they're transmembrane proteins. They're involved in the transport. Then we went on to transport proteins. There are two types of transport proteins. There's channel and there's carrier. We did channel right here, where a polar hydrophilic channel goes through a non-polar protein. Then there was a gated channel where the gate opens up to allow the ions to travel. Then we went on to carrier proteins. There were two types of carrier proteins. The first one were simple carrier proteins, which showed a conformational change. Now these carrier proteins used diffusion, as I stated right here. They use diffusion, and that's how they bring it using a peristalsis type of journey into the cell, right? And then there is pumps. These pumps are used when active transport is required, and we've stated how pumps act. So that was it for today.